The idea that you can't change who you are is used in cheesy films to encourage the protagonist to uphold their duties, and in real life by unpleasant people who want to justify not changing their behaviour. But in biology, it's recently starting to prove untrue. Everyone watching this video started life as a single cell, as did everyone not watching this video. This single cell, this single fertilised egg, successfully turned into a person, which means that it contained the capacity to turn into any cell type. This is called pluripotency. Now, the first step towards becoming a person was for the cell to divide and produce a ball of cells which were identical to each other, which were all pluripotent. And this is what people refer to when they talk about embryonic stem cells. The next step in becoming a person was for these cells to differentiate from each other by switching on and off different genes. Every cell in your body does contain the same set of genes, but they're not all switched on in every cell, which makes intuitive sense if you think about it. If all of your cells were doing the same thing, you'd be a homogeneous blob. This process of differentiation can be thought of as a series of branching one-way pathways. Cells can't reverse back up the pathway, and they can't hop from one pathway to another once they've taken a fork. But not every cell will follow its differentiation pathway to its endpoint. Some of them will halt at a fork in the road. In adults, we possess reservoirs of cells that can turn into several different cell types. For example, bone marrow stem cells can turn into any type of blood cell, which is the basis of the bone marrow transfusion. There has been a lot of interest in the last 10 years in using these cells to repair and regenerate tissue, but they do have limitations. As I said, they're restricted in the number of cell types they could turn into. They're few in number, and they can be difficult to obtain. This is where induced pluripotent stem cells come in, or iPS cells. iPS cells are adult cells, fully differentiated adult cells, which have had pluripotency switched back on by introducing four genes for pluripotency, using a virus. And when these iPS cells produced in a mouse were introduced into mouse embryos, they integrated fully successfully, and the animals that were born were perfectly healthy, perfectly viable. The embryonic cells didn't recognise that there was anything different about the iPS cells. So this could be a way of combining the pluripotency of embryonic stem cells with the more ethically attractive option of cells obtained from a consenting, sentient adult donor. It is early days, as I said, but so far the iPS cells have been successfully produced from stomach, liver, urinary tract. The first ones were produced from skin. So iPS cells are proof positive that a leopard can change its spots, but depending on how literal you want to be with that, you might need to let it take some time and perform some genetic manipulation.